Okay, so very excited today to have with us John Tsang from Yale, and he is going to talk about how the Immunome Project and IMH may further the understanding of the origins of type 1 diabetes. Um, John is a systems immunologist, a computational biologist, and an engineer who serves as the Anthony N. Brady Professor of Immunobiology and Biomedical Engineering at Yale, a Chan Zuckerberg Biohub investigator, the Yale lead of the CZ Biohub New York, and the founding director of Yale's Center for Systems and Engineering Immunology. Trained at Harvard, PhD in biophysics, and University of Waterloo, he investigates why human immune responses vary widely across individuals using machine learning, quantitative modeling, high-dimensional immune monitoring, and experimental systems. His group aims to develop predictive immune cell engineering tools to sense tissue states and enable early disease detection. A former senior investigator at NIH and co-director of the Trans NIH Center for Human Immunology, he received multiple NIH NIAID merit awards and advises major national and international immunology programs while contributing broadly to the fields of systems and quantitative immunology. I'm going to drop two sort of interesting papers that pertain to his work into the chat now, so you guys have them at your fingertips. And really appreciate John taking time out of the busy schedule to walk us through what's going on and give us some updates on this exciting set of tools.
Okay, great. Thanks, Monica, for having me. Very excited to be here, and I'll keep I have kept the sort of talk fairly high level general to touch on sort of multiple aspects, highlighting this notion and concept of human immune set point and immune health. And if you have any questions along the way, feel free to interrupt or we can discuss as well. So some of the details you can actually find in this recent review paper of ours, and especially pertaining to this concept of set point and immune health, and how machine learning and AI and high dimensional immune profiling are really helping us to push these forward. So some of you may appreciate that variability, both across individuals and across human populations. It's really a hallmark of the human immune system, and in terms of immune response outcomes. So you see that in the context of immunotherapy response, for example, in cancer, anti-PD-1, PD-L1, or CTLA-4, those responses are typically far from 100%. So you get anywhere from 30 to 50% of the individuals respond. And also a variable percent of those individuals also can have adverse events. Actually, that turned out to be quite an interesting scientific sort of arena for understanding autoimmunity as well, because a lot of the patients actually mount autoreactive responses. And you see that in the response also in terms of vaccines and infections. And you can actually see hundreds of folds of differences in immunogenicity. In this case, I meant the antibodies increases as a result of vaccination. And you see that hundreds of fold differences across individuals. You also see that typically correlate with age. You get attenuated responses when one gets older. And of course, you see this in autoimmunity in a variety of outcome variables. For example, in terms of disease activity in lupus patients, some patients flare a lot more than others, and also the flare intensity can be highly variable, and also the type, the quality of the quality of the type kind of flares. For example, whether it involves certain myeloid cell populations or B cell populations in the blood, those are also highly variable across patients. And the question we and others have been asking over the years is, can we predict these responses in humans? And can we, based on those kinds of signals, can we start to uncover determinants and on their shared mechanisms across these seemingly diverse heterogeneous scenarios? So that could lead us to start to define, for example, endotypes, meaning classifying patients and individuals based on the underlying mechanisms and molecular biomarkers, rather than just based on clinical symptoms, and which typically can fail in terms of identifying the right treatment regimens, for example, or even how to treat a certain individual. So our journey started actually more than a decade ago, when we used a vaccine as a way to probe these differences across people. So at the time, we did a study where we enrolled a bunch of healthy individuals. We gave them a flu shot, and we took sort of two baseline time points before vaccination. So that allowed us actually to discover that the baseline of individuals can be one, quite unique to that individual across a variety of variables like immune cell population, states, and so on. And second, those baselines actually, at the time, quite surprising after we integrated data from both the pre-vaccination time points as well as post, we found that the baseline, the differences I mentioned across individuals that are quite stable over the course of months, actually can predict the antibody response to the vaccine beyond what can be provided by variables like age, biological sex, and also antigen-specific, in this case, flu-specific immunity, right? So we actually are able to reduce that signature to actually only 10 genes in the blood. So that's moving towards having cheaper and easier to evaluate surrogate biomarkers. And that those 10 genes allow us to measure, basically compute a score. And then we tested that in a number of independent cohorts involving influenza as well as yellow fever, so which is a different vaccine. And in general, we found that the same signal, without any further training in these cohorts, can delineate the high versus low responders, except in one set of cohorts from one particular location where we had a study plan to test it, but that was one out of the multiple other ones we tested that didn't work out. Um, so so here's one concept I want to get across that actually touches on autoimmunity. Um, at the time, it was quite I was quite fascinated by actually still fascinated by lupus. Actually, how this is actually one patient tracked over time by my colleague Virginia Pasquale, and this patient would have variable disease flares, as you can see over time. So Virginia's group sort of pioneers this approach of looking at the transcripts in the blood, and based on that, they cluster their pediatric lupus patients into 7 groups. And these groups are defined by different characteristics or different fingerprints, mostly driven by what they look like transcriptionally in the blood during flares. So 3 of these groups caught my attention because whenever they flare up, these patients would have what's called a plasmablast signature. So these come from B cells that are making antibodies. And what's interesting is that when you vaccinate a healthy person, typically by day 7 to about day 14, you see a acute peak in the blood that are also driven by plasmablasts. So then we thought, well, this could be interesting because the same baseline signatures that we detected that can predict vaccine responses in healthy individuals may actually be helpful also in predicting flares. So, for example, if you evaluate a particular patient during their clinically quiescent period, would that contain information about the strength or the intensity of those flares? So we tested that in this paper, and turned out that as we had hypothesized, these patients having these plasmablast flares, there's indeed a significant correlation between their baseline state as measured by our vaccine predictors, actually the set point I mentioned, and the intensity of this plasmablast signal, basically. When we apply that to all the other patient groups with different kinds of flaring characteristics, there's absolutely no correlation. So it's very subtype specific for this set of lupus patients. So you may ask, what exactly is this signal that I mentioned, right? That can be captured by those 10 transcripts that I mentioned. So by 2018 or so, we started to titrate a panel. This is a SciSeq panel. This is before all these lyophilized, off-the-shelf, convenient panels became available. So we look at 82 cell surface proteins together with transcriptome in single cells. And then we contrasted the high versus the low responders in our original vaccination cohort. And what we found was that the signal that we saw initially and what we found computationally by finding this 10 transcript surrogate actually reflected a network of cell states. So in high responders, these are, again, clinically healthy people, but they actually have more activated PDCs. And these cells presumably are making more interferons because, one, they're major interferon producers, and they're implicated in autoimmunity, for example, in lupus also. And the holes in blood in these high responders, they have an elevation of interferon signatures. And then there's also elevations in the frequency of these activated B cells, together with this CD40 ligand response signature in the T cells. And interestingly, these are coupled. They are correlated. So if a person has an elevation in the state of one cell, particular cell type, we also see the corresponding elevation in the other ones, basically, suggesting that there's an underlying circuitry, the communication among these cells that basically kept them at this kind of high state. And then later on, we found that some of these baseline or set point signals, they actually resemble the action of adjuvants. So adjuvants are typically synthetic perturbations or agents that can enhance the immune response of an individual, for example, to vaccines. So when we look at this particular adjuvant in this paper, ASO3, we saw that if you look at the unique immunological changes induced by the adjuvant quickly following vaccination, that same signal actually can be naturally variable in the human population. So some of us, without any perturbations known, are already sitting at a stable, higher set point state, resembling the unique action of these adjuvants. And that's one reason why these individuals can respond higher to vaccination. So in general, the set point concept I introduced, at a broader sense, it's what we call these personal immune states, and they're obviously very complex. And if you just tally up the number of possibility or possible configurations, the number is huge. But of course, not all configurations are possible or even are present in humans, because there's a lot of constraints and there's evolutionary selection and so on, right? So there's lots of different factors that can shape these immune states, including some of the like genetics and biological sex. I call those intrinsic variables. There are also more complicated composite variables. For example, age. It's a complex composite variable because it reflects many different combinations of things, right? There's developmental progression and aging, but there's also cumulative exposure because of the environment. So all of those things are reflected off with age associations and so on. And if you think about there are two major timescales that contribute to these personal immune states. One, it's over long evolutionary timescales that shape genetics. For example, HLA genes and SNPs around many of these genes can shape their expression in states in immune cells and non-immune cells. But perhaps, especially the immune system, which is this monitoring system that respond to the environment and try to adapt, even over the relatively short time scale compared to evolution, right? Over the lifespan of an individual, that's a major shaper of the status of the immune system. Not only antigen-specific states in terms of clonal expansion of T and B cells, but also in terms of epigenetic regulation within specific cell types, as well as interaction, as I alluded to earlier, among the cells. And those interactions on their own can also shape and actually stabilize certain cell states, and those can persist over variable timescales. And so the key question is, if these are shaped both by the environment and by genes, and as well as different across individuals, they ought to be very informative and provide this big window into health and disease. And so that's what we call by things like immune health. And the question is, to what extent is our immune system capable of responding to various stress signals? That's one, right? And second is, can we utilize the immune system as a window, in the sense that it's such a good sensor, it has to be able to read out and get information within our own body, as well as, of course, outside our body in terms of threats. So therefore, it likely it's a very informative sort of point through which we can read off not only what's going on in the immune system per se, but actually what's going on in our entire body. So technologies, especially advances in the past decade or so, really now enable us to look at the state of the immune system fairly comprehensively, and the costs are also dropping. So in a typical experiment we do in humans these days, for example, we can take a sample. It could be a drop of blood. It could even be specific tissues that we can biopsy. We typically get about a billion numbers per sample per time point per person. And these involve both fixed states like genetics, but also these things that could evolve and shift, for example, cell states and TCR repertoire. You can also take the cells out and generate quite a bit of data from them, right? So in a single person, you now get a huge vector of numbers. The question is, what do they mean? It's of course the key question. So I mentioned already that these set points that we have seen so far, they typically exhibit stable characteristics within months. When you measure the same person again, you can actually find that these parameters are fingerprints in the sense that while they fluctuate over time, the differences across individuals are much greater. While this example at the bottom is actually more rare. More often you find that people are stably different, right? So now I want to show you another example where this actually concept of stability is actually helpful by helping us to tease apart different kinds of signals. I mentioned earlier that checkpoint blockade can actually induce autoimmunity in cancer patients. So we got curious and decided to do a pilot. So this pilot study, we were working with NCI colleagues to look at patients with thymoma. So thymoma is interesting. So these patients, when they get, in this case, anti-PD-L1, typically about half of them would have myositis. While half of them are fine, no adverse events, and they are all good responders. So 100% of them have good anti-tumor responses in this particular set of people. So that the question about this notion of can we predict who may get IRAE, and also can we potentially even mitigate IRAE? It's always clinicians always bother by one potential issue, which is if you mitigate IRAE, to what extent are you going to also dampen the anti-tumor response? Because they worry that the anti-tumor response is coupled to the IRAE. So therefore, I won't go into the sort of the computational details. We come up with an approach to basically say we can come up with signals that likely are corresponding to anti-tumor responses. For example, when you look at high and low, or maybe IRAE versus no IRAE individuals, they both respond in the same way before and after therapy, right? You may also find that there are differences that are coupled to the response, but yet in the IRAE patients, they, let's say, have a higher signal than those who don't, right? These are also not desirable because they may still be coupled to the anti-tumor response. Instead, what we ask is, can we actually take away all of such potential signals? And are there any signals that are already different? So back to the set point concept, right? That the patients are already different to begin with at the baseline. So the therapy doesn't actually change their states. And these could actually represent meaningful and maybe targetable to mitigate this kind of IRAE response. So that's what we look at, and we found that yes, indeed, there's actually multiple signals in different cells. So this is looking at cells in the blood, different cell types, and these are the states of those cells. When it's red, it means it's elevated in the IRAE patients. So one prominent signal actually comes from the CD8 TAMR cells, where the genes downstream of mTOR signaling, it's actually elevated in these patients, as you can see. And importantly, it's not just the TAMR cells. It's actually, again, back to the concept of the couple signal. It's really signal across multiple cell types that are correlated. So you can see increased inflammatory states in dendritic cells, for example, and these are correlated with the TAMR signal I mentioned, right? And then Matt, then actually, this is an older slide. He has now done this even more extensively. He has looked at multiple independent cohorts and asked, can the same signal in different patients with different tumors actually predict these IRAE responses? And the answer seems to be yes so far. And he also had looked at tissue data to look at tumor-infiltrated lymphocytes versus like T cells that are just sitting in different tissues, and found that these signals tend to be significantly more enriched in the tissues, but not in the tumors, not in the TIL, that infiltrated tumors, suggesting again these signals may be quite specific in mediators that may induce the adverse autoreactive reaction rather than the anti-tumor response itself, basically. So now getting maybe closer to type 1 diabetes, it's thinking about kids. It's how are these antigen agnostic set point that I mentioned established, right? So you can kind of think of two extreme scenarios. One is where they're established early, potentially maybe even by genetics, and then they maintain their states over long periods of time. Although that's actually quite intriguing when you think about it, because we are constantly facing with signals from the external environment and development. So imagine if something is it's set fairly early on, how are they being maintained over time, right? The other one would be no, they're the typical paradigm is that they're age associated. So early life and early development, and over the first decade of life, these things are being shifted and changed. And then some of them can still be modulated by the environment when we become adults, right? So the answer to those questions, we work with Eva Harris and leverage a fantastic cohort she has developed over the years in Nicaragua, where they longitudinally follow kids starting from about 6 months of life all the way up, and they have longitudinal sample collected from these subjects. So based on those, we designed a study where we follow, we basically put together multiple subcohorts and follow these individuals longitudinally over time for at least 6 years. So you can see here that we have one subcohort going from about 6 months to 6 years, and then so on and so forth, and so allowing us to bridge from 6 months all the way to about 14 years of age. From there, we want to ask this question again, right? Where does individuality come from? When does is it prevalent already in early life? Like these intersubject differences, basically, right? So surprisingly, actually, so this is looking at all the transcripts from immune cells after accounting for cell frequencies initially. We found actually that because we have multiple time points per individual, we can reliably estimate the extent by which the variation or the differences in gene expression, in this case, can be attributed to subject differences versus biological sex versus age. So as I mentioned, the prevailing paradigm is that age, it's a major dominant driver of how the state of the immune system may shift, right? But in fact, we found that most things are actually stable. They can be quite different already from person to person. While the contribution of biological sex and age, they're relatively small compared to these intersubject level differences. So here's an example of what I meant. So here's each line is a person, and you can see that independent of which subcohort you look at, like at different starting ages, the level of this particular gene, it's stable within a single person over time. But you can see that on the y-axis, you can see these variations across people to be the dominant difference, basically, right? So then back to the point about nature versus nurture. Are these mostly genetics, or these are actually maybe very early environmental influences that sets those set points? And so there's various studies in the literature, for example, based on twins, that typically estimated that the immune system in particular can progress from initially being more shaped by genes, genetics, as slightly later in life, things are becoming more and more environmentally driven, which makes sense. So to look at this more closely, to really look at like where do those signals come from in terms of cells, we had done a follow-up study, but now profiling every single sample with single cells analyses. And now we have followed some people for even over 10 years following a similar design. And from here, we can start to look at within individual cell types, where do these temporally stable intersubject variations come from? So for example, I'm highlighting sort of two clusters. So these colors, by the way, they indicate sort of how different these genes, so each column is a gene, are from subject to subject and how stable they are. So you can see that in monocytes, there's a set of genes that are fairly temporally stable. They're explained by intersubject variations. And here, actually, we're showing pooling these numbers from these heritability studies. So showing that in monocytes, about 30% of these genes have been deemed heritable based on twin studies. The rest, the effects of genetics are small, relatively small, actually. And this is even more pronounced in genes that are particularly having high intersubject variation in T cells. You can see that only about 17% of them are deemed heritable. The rest are not, basically. And these are typically involved in T cell activation, these genes. And in the monocytes, they are typically involved in suppressors, actually, negative regulators of inflammatory signaling. Um, so you could sort of maybe surmise that these early regulators, maybe of the threshold by which these cells may get activated, are established fairly early in life. And once they're established, they can be temporally quite stable within individuals over time. Some of those are definitely set by genetics with strong effects, but many of those are likely also shaped really early on by environments. So we look at this a little bit more in older US adult cohorts and ask, do the same genes that show this kind of high intersubject variation and stability in the pediatric subjects in Nicaragua, how do they behave in adults, especially in the US? So we took advantage of this public data set and asked that question. And we saw that the intersubject variability actually drops in monocytes and B cells when you look at these adult cohorts. But what's interesting, it's in these NK and CD8, CD4 cells, they remain highly variable across subjects. In other words, their sort of stability may be maintained all the way into older ages. Now, we haven't looked at these people, of course, from when they were young all the way to the way they're old, so it's not the same people. We're looking at two sets of different cohorts, one adult, but it's interesting to see the parallel in terms of the stability. So you can see that when you look at monocytes, these are the red dots I showed in the previous heat map. You can see that by the time we look at adults, they're actually lower in their intersubject variability. While if you look at the T cells, the CD8s and the young and younger adults and the older, they're more or less consistent, or maybe they're even more stable in the old. So there's, I think, a likely substantial regulatory structure to help maintain these individualistic set point states within individuals. We're still kind of trying to think about why and how and why is this important. Once you sort of set a set point, why is it important to maintain these things? So and then we try to ask, are these genes really functionally relevant and have potential relevance if you actually perturb them? And I won't go into the details, but when we look at genes implicated or signatures of autoimmune diseases, and also genes at baseline that predict vaccine responses, especially when you look at the genes that are present in both, you can start to see that they are more enriched for those genes from the pediatric cohorts that are more explained by differences across different subjects, basically. And they also tend to be driven by genes with lower heritability. So the genes with higher heritability, meaning genetics, it's more likely shaping it. There's not a lot of huge enrichment across the board, but the uptick here, it's mostly driven by these genes with lower heritability, basically. Now you would ask, like, where do we see these six developmental sort of signals where it's not the change in average, but actually these individuality signals actually come later in life. So by that, I mean, initially, if you look at two individuals, the red and the blue, they're the same, they look about the same. But then later on, because of a developmental change or an environmental exposure, the red and the blue individual are starting to diverge. They're now becoming two different looking individuals, right? And the opposite can also be true. You can imagine initially they're different, let's say because of genetics. And then later on, they are now becoming more similar. And the ones I've shown you so far, they typically exhibit what we call persistent signals. So they're over time, they maintain their differences. So while they fluctuate, those are small compared to the differences, right? So we were surprising to see this pattern over multiple years, to be honest, in the kids. So here we actually try to understand where do we find these increasing, decreasing VES, and we found that interferon is actually a good example. Initially, interferon signatures are all dropping across all individuals. So on average, they're all dropping. But by 4 or 5 years of age, they start to become more individualistic, basically, likely because of different exposures and to infectious diseases. So some would maintain sort of elevated interferon signature levels, and one would have lower ones, right? So here I want to highlight also maybe for you as particularly interested, this is IAB, B cell, and IAA signature. So these were signals found in a TEDdy study, where they try to ask what are some of the early signatures that can predict the emergence of autoantibodies in kids who develop type 1 diabetes. So you can see that these, for example, so here's looking at the relative sort of stability of the genes in those signatures. And you can see that on the left here, so as you would expect, because this is a B cell signal, more of the intersubject variations are in the B cells, right? But you can also see that some of them, green are the increasing, some of the genes within those signatures, they have this kind of increasing individuality. So meaning they actually emerge over time as well. So some of those may actually be potentially relevant for the development of diabetes, maybe at least the emergence of increases in risk. But if you look at this IAA signature, I think it was found in T cells. So you see very high intersubject variation in the bulk data, RNA-seq data. And specifically, when you look at the single cell data, it's localized to NK cells and CD8 memory cells, basically. So it's already quite stable to begin with. So again, maybe predisposition is really, really an early event in these kids, basically. How much time do I have, Monica? I have still the IHM kind of
Sure.
concept to
Yeah.
highlight.
No, I'd love to hear whatever you can, and people stay on. We also record this for sharing with our community later.
Okay. Yeah. So back to the point about immune health. I want to say upfront that it typically when we think about immune health, we tend to, well, what exactly does it mean to be healthy, and do we need to study the healthy people and understand their ranges, and try to understand getting the right healthy baseline that allow us to study disease and so on. But here's an example where I think they're all intricately linked, because if we don't have enough observations and examples about how an individual can become unhealthy, our notion of healthy is actually would be missing negative examples, right? Basically. So to learn what it means to be healthy and to quantify what it means to be healthy, we need to learn from lots of examples of how someone can become unhealthy. So this is one of the questions driving us to understand these monogenic diseases. So at the start of this project, that was more than a decade ago, the question is, are there common deviations from health that we can detect from these very diverse patients with monogenic lesions? So typically, these patients suffer from a highly penetrant, high effect mutation that impact, for example, the STAT3 or STAT1 gene, PI3K. So these are key signaling molecules and sensors in the immune system. So, and the second question is, can we utilize these as human knockouts and knock-ins to then understand better on the underlying communications and wiring of the immune system? And so we thought that these would really provide good negative cases for us to learn about common deviations from health, right? And whether they exist or not, basically. So this is a heroic work led by Rachel Sparks, a former clinical fellow in the lab, and together with Neha Bansal, they together collected now more than 400 samples from 300 patients and matching controls. And for some of those, we have longitudinal assessments. So therefore, again, we can ask temporal stability. And from these, we have generated lots of data from these patients. I won't belabor on that. So one idea that we've been exploring, it's this idea of, can we define a coordinate space such that we can actually, in a unbiased way, without knowing which patient and who is whom, actually put these individuals on this coordinate space and be able to uncover in a fairly unbiased way things like endotypes and the differences across patients. Of course, the problem, it's the data we collected, it's very high dimensional. So the classic approach of doing this, of putting these onto these high dimensional spaces, can be challenging. But of course, that's where machine learning and modern AI kind of concept comes in, right? So you heard a lot about AI these days, and if you look at underlying the hood of these approaches, I would say all of them do some form of this kind of compression or representation, where they come up with what's called a new lower dimensional representation or manifold of the data, right? So this is a very simple example illustrating the concept. So imagine you got a complex object living in 3D space. So you have each point is described by 3 numbers. But you can see that this particular object is fairly complex, right? It's got a complex shape. And you can computerize, vectorize that shape, but in fact, once you kind of represent this object in a different way, you roll it out, and it becomes this flat 2D object. In fact, you would argue that it's actually just a 1D object, because most of the variation, it goes along the x-axis, not the y-axis, right? So it's actually a 1D object. So if my goal is to say, if I give you a point, can you predict the color of the point? It becomes a very easy problem when you represent the structure in this kind of way, right? So you can basically say, if the number is greater than or less than a particular set of numbers, you can call it blue or cyan or green or yellow, right? So it becomes an easy problem. So that's what a lot of AI is doing these days, is take lots of data, try to come up with these kinds of useful representations, and then take this representation and feed it into downstream things that we want to do. It could be predicting a patient's response to a therapy and so on, right? So we have done basically the same thing in this pan-monogenic exercise. As we took these individuals, we look at the different kinds of data that we have generated from them, transcriptional data, cell data, protein data, and so on. And then we come up with a way to come up with a coordinate space and put these folks on them. And then we try to understand what these axes are telling us. And then we also have done some what's called supervised exercises. So on the left, it's unsupervised, because we actually look at these individuals without any labels. So we don't know who has what disease and what kind of symptoms they have, nothing. So we just want to look at them using their molecular cellular data in an unbiased way. While on the right, we're asking, can we find common deviations from health across all of these people, right? All different kinds of patients, basically. And then computing an index of saying, if you have one single person coming in, can we compute the probability that that person is sick, healthy, maybe along anywhere in between, basically. So what was quite surprising, it's when we look at this space, especially the very first dimension in that space, it's actually highly correlated with what we computed on the right, when we know who's sick and who has disease. And this is surprising, because we have tried this even when we only feed in the patients, no healthy controls, whatever whatsoever. You basically still come up with the same space. And turn out that this is a very robust axis of quantifying people's immune states. And we thought that it's actually appropriate to call this an immune health metric, because one, they can clearly tell, just like this supervised model is shown, the probability of someone being so-called clinically healthy, right? And second, because it's continuous, it actually starts to delineate the different shades of health. By health here, I don't just mean how good is my immune system, although it's correlated with that. For example, this is I'll show you some data on that. But also it's about what the immune system is sensing. It's almost like a downstream signal that whenever there's a perturbation or deviation in our body, this set of signals gets changed, and therefore the immune system can sense it, basically. So you can see that I'm plotting here. Each dot is a subject, a patient, and on the left are the different categories of them, the initial genetic diseases categories they are assigned to, basically, right? So you can see that there's extensive heterogeneity within each one of these groups. At the very top, the healthy, clinically healthy people, right? On here are people with typically with more severe disease. So for example, CGD patients and CTLA-4 mutation. And what's interesting is this extensive variation in the clinically healthy population. Some of them actually overlap with people with disease. So the question is, are they on their way towards disease? So then we decided to look at independent cohorts and found that despite the fact that we don't have any aging signal in our cohort, because for example, the healthy controls and the disease patients are all fully matched. But when we look at folks, clinically healthy people across the lifespan, we saw a gradual average decrease, right, in the immune health metric score of these individuals. But of course, there's huge variation. So even for these 80-year-olds, for example, some have an immune health metric score look like a 45-year-old average, right? And some are much lower. So I've since worked with also an independent set of cohorts with colleagues from Japan, where they follow people who progress on to become, some of them become centenarians actually. And there, the preliminary results are also very, very interesting. So one, they confirm that this differences, so you get a bunch of 80-year-olds and you look at the high versus the low ones. In the Framingham cohort, my colleague Professor Cartry has looked at this IHM metric, and they found that this metric actually predicted all-cause mortality in that cohort. And then we confirmed that in the Japanese cohort as well. And more interestingly, there may actually be signals about not only mortality, but also about the quality of life and the lifespan, so to speak. So that's a signal that we're still checking right now. But interestingly, this actually predicts forward in time, right, in both cases. And also, as you can see here, that when we look at a different cohort of patients experiencing heart issues, heart failure issues, this metric tracks that based on the timing of the onset as well as the severity of the disease. And also, this is showing you how it tracks like responses. These are RA patients, either responders or non-responders to therapy. And again, you can see that it's tracking the status of these individuals. And we also have data showing that, especially in older individuals, this metric predicts their vaccine response outcomes. So when you look at people who are 65 and above, their response to influenza vaccination at the baseline, it's correlated. So the baseline status of the IHM is correlated with their influenza vaccine responses. So here's just a sort of a figure showing the concept behind what this is telling us, right? So we know pathology typically from symptoms, right? So these are far out from optimal. And we typically don't know exactly what we mean and how to quantify healthy either. But I think with these baseline immune states and this concept of immune health metric, we'll start to, I think, to be able to start to point individuals along the spectrum, basically. There may be individuals who may still be healthy, and these could include autoimmune patients who are progressing onto autoimmunity, for example. And therefore, if we can really start to quantify where they are and what stage they may be at, it potentially could help us intervene and modulate and start to push people back, right? And also, I want to also clarify that I don't mean to simplify too much. This immune health metric, while it's a very, very strong common signal, basically, across all of these different perturbations. We have looked at autoimmune patients and different kinds of patients way beyond the monogenic cohorts. It's still the same, but I don't mean to say that the immune system can be quantified in one dimension. I don't want that to to be a misunderstanding. I think that's just a first glimpse into how, if you look at the shared signals, we can learn about these foundational states that are really relevant to human health. And I think we still need higher dimensions. It's not going to be tens and thousands, I think, but it's also not going to be one. But the goal is to be able to delineate and develop these approaches, I think, to quantify the state of the individual, basically. Um, so so yeah, we've been also interested in this concept of for type 1D, what are the set points, basically, right? Are there pre-disease signals that one could study? To do this in a general population, obviously it's difficult because it's the prevalence rate is still low, so therefore you need to look at a large number of people. So we are in sort of discussion and collaboration with some colleagues where at risk, like babies, for example, born from at risk parents are enrolled. So looking at those is one avenue. We also have been working closely with my colleague Kevin Harrell at Yale, where and Kevin and his colleagues over the years have developed this anti-CD3 antibody therapy that delays the onset of type 1 diabetes. So we're starting to look at samples both from earlier trials as well as more recently to sort of see how these may not only delay but maybe even mitigate disease onset in certain patients. So we've been generating data, tracking some of these folks over time to look at both what we call as induced signatures. So here's one example where the anti-CD3 induce a shift, and those shifts can be distinct between responders and non-responders. So this is just a glimpse of what potential signals we could find, and we're of course interested in looking at the baseline. So final point, it's as Monica mentioned, we're also pushing forward on a project called the Human Immunome Project. This is closely related to the idea of understanding these set points in immune health, but really can we do this across the globe and not missing the very extensive variation and signals, and those are likely very, very important to help us understand better what these immune states, what are they, how do they link to specific outcomes, and can we start to develop models that can actually be applicable not only to the study population in the US or EU, but actually can be used in a variety of contexts, right? So given this motivation, so we thought that the time is really ripe to launch this project, to do this kind of work and look at applying the same approaches in terms of study design, longitudinal tracking, in terms of sample collection and processing and data generation across the globe together. So we have now a partner network of 10 sites going to be 11. This spanned the globe, as you can see, and we just had our first Benbury meeting at Cold Spring Harbor Labs, discussing the plans for the first two phases of this enormous project. And we're all very, very excited, and we're now coming together, hashing out the study protocol and the technological approaches to put this together. So with that, I want to really thank your attention and invitation. Happy to discuss and take questions.
Thanks so much, John. That was really a deep dive into what's going on there with your team and your collaborators. I want to open it up to other people in the audience if you'd like to ask a question.
I can start. Hey, John.
Hey,
Thanks so much for the fantastic talk. So I have two questions. One, what are your thoughts on incorporating, TC or best ways to incorporate TCR, BCR repertoires in that? Because as you can imagine, it's a weird scenario where there is way too much sort of intra-individual variability longitudinally and at a given time point, and inter-individual variability as well.
Yeah.
And then from your pediatric cohort, are you able to sort of travel back in time and infer sort of infection history with reoviruses or things that kids normally get in a normal sort of healthy early childhood?
Yeah, those are awesome questions. So the maybe I'll take the second one first. The second one is, I think, for recent infections, things like interferons and things like that are actually fairly informative, because the relactation time scale can be can be longer, and we're still kind of quantifying that to see the extent by which we can tell something. Now, those are non-specific, as you can imagine, right? For specific ones, we have some preliminary data on looking at the antibody reactivity profiles. So you can now, like for example, the phage display and FIB-seq and technologies like that to look at the profile of your antibodies, at least in terms of linear motifs. And those can be quite informative in terms of the exposure history. Now, again, you can't quite tell the sort of time, right? Like whether it's early versus like you acquire it later. But for example, using vaccination as a positive control, you can indeed kind of see the emergence of the antibodies because of the vaccine. So you can see that those are changing because of vaccination exposures, and those are controlled. So using those, we're getting, I think, optimistic that yeah, that kind of approach at least can give us a sense of at least checking off like 1 or 0, like
Right.
has this person been exposed? But when quantitatively, those are trickier, I think.
Right.
Now, the repertoire, I think, is fascinating because, as you know better than I do, the amount of private sequences, it's dominating, right? So that I think it's until the point when we have really strong, I wouldn't say even specificity, because I think that's a tricky concept. Until we have like reasonable predictions on the likelihood binding profile, for example, or the recognition profile of these receptors, it's very hard to bridge those ones. So therefore, we're going to rely on public ones, right? Public clones, and those are interesting. So did you see the recent maybe Scott Boyle's paper, for
Yeah.
example?
The MAL ID.
Right, now ID, right? So there's seems to be signal. Not in terms of infection; those are relatively easier, I think,
Right.
because you can see expansion
Yeah,
of
right.
clones. But even specific diseases, there seems to be some pattern. I think it remains to be seen to see how generalizable some of those are. But I think it's exciting that some of those public clones, and we're sort of excited to kind of look at those and ask, like, how do they link to some of these specific molecular cellular signals that we have been discussing. Yeah.
Great.
But
Thank you.
but I don't, yeah, but otherwise, yeah, public, hopefully seems like there's enough signal already. Interesting. Yeah.
Yeah, just a little quick plug is that the Sugar Science did conduct and complete and in progress, or this project's in progress. We put together a community build of a T1D specific TCR BCR data repo, and that's housed on Air Data Commons and accessible through iReceptor. So it's there, and it has quite a few sequences. We have a monthly working group, and Scott Boyd did just speak to us at the end of the year. So that's very cool. I had a couple of questions for you.
hmm
One is, you know, did you guys you know, uh, there's this personal immune set point that predicts the outcomes you alluded to in the very beginning of the talk, and you said there were 10 genes that you pulled out. Do any of these genes correlate with genes selected for by the sort of the Black Death survivors like the ERAP2 and all those others, or no? Because that was sort of floating around that hypothesis for a while, a couple years back, that those that were more susceptible to autoimmunity might be those who survived the Black
Got
Death.
it. I don't think the genes are in the signature, but it doesn't mean that the signal are not correlated. So I think we could take a look if you guys have a very specific like signature and hypotheses. We could
Mhm.
actually go back and look and see. Now we have multiple of these set point signals. We could ask like which one and which combinations of those may be linked to the one you mentioned. Yeah.
All right. Well, we'll talk later about that. Um, so yeah, so I had a question for you, like, Um, have you ever applied or considered applying the IHM to longitudinal T1D cohorts? And as I alluded to, like off camera and just dropped into the chat, there's this really great new paper from Johnny Ludwigson and a University of Florida group, including Eric Triplett, who was on the call earlier. Um, do you think like they're saying basically it's their pregnancy in the placental tissue and et cetera, at birth in some of these individuals who go on to develop type 1 diabetes. So, like, if you guys had access to those samples, do you think you could apply IHM to longitudinal T1D cohorts of that group?
Absolutely. Yeah, absolutely. So we have been thinking about that in the context of the anti-CD3 trials, as well as in these like early life enrolling them at risk kind of studies, some in Australia, and then of course Teddy, it's the one based in the US and so on. So I think absolutely, I think both in terms of testing very specific ones that we have, as
Mhm.
well as depending on the power, really cutting across these ones to uncover potentially new ones, basically. And I think also the hypothesis that stochasticity may be involved, and to what extent is that involved? It's also, I think, an important question. So
Yeah.
maybe indeed, yes, I think environment and other triggers may shape the baseline propensity of the system to cross certain thresholds. So we actually had worked on a paper with Ron Germain, Harry Wong, for example, where we're trying to ask this very basic questions of there's multiple checkpoints for prevent what the body is trying to, of course, not allowing these autoreactive cells to become active, basically. And
Mm.
but what exactly are controlling these thresholds? And we all think that, of course, these are highly nonlinear thresholds, right? So there's multiple things going on. If one thing goes wrong, typically it doesn't. It's so there's multiple things, and or is it the regulation of the threshold itself that's being disrupted, right? So you may have a very high threshold, therefore fluctuations or changes in the system will never get to that bar. But if that gets lower, for example, then the regular fluctuations you see would not be buffered anymore, for example, right? Because I,
Mhm.
I I think the
Yeah.
system has to be able to cope with unanticipated stochastic variations, and so therefore certain thresholds has to be set fairly high so that you can buffer them. And I think the breakdown in tolerance, so-called breaking down in tolerance, has to do with regulations in those signals, maybe even more than specifically there's a trigger, let's say a virus, EBV or something, that boom, once you see that signal, you're now going to develop on the trajectory to develop type diabetes. And that's
Yeah.
also
Well,
why
there's different endotypes, as it were, right? Like there's a cohort that develops at younger, sort of what I think 5 and under. And
Mhm,
then there's the sort of the teenage group, and then there's the adult group. So there's different progression. I mean, some people talk about it in a relapsing, remitting
mhm, mhm,
um,
mhm,
structure, that this is how it's sort of like, you get the one hit, you recover, and then next, et cetera. But I think that the way you're looking at things is going to really um, open up the door to understanding the circuitry, which I think is
mhm.
pretty
Yeah,
important.
yeah. No, I would
Obviously,
love to follow up on the potential cohorts you mentioned, and also some of these more theoretical ideas that and how to explore them experimentally. I think
yeah,
would be
we can do that. Maybe we'll have you come to our working group. Here's one last question. Great talk. You mentioned that the baseline high responder signature in healthy people driven by PDC type 1 interferon and B cell networks overlaps with signatures seen in the SLE patients during plasmablast associated flares. My question is about the specificity of these inflammatory states.
Mhm,
Does this viral ready high responder baseline also align with the mTOR driven signatures you've recently linked to IRAEs or immune related adverse events in cancer therapy? Or are vaccine high responders and IRAE prone patients biologically distinct types of hyper responders?
mhm, mhm, mhm. Yeah, good question. So they are partially overlapping, but depending on which healthy cohorts, clinically healthy cohorts we look at, the correlation can be variable. Basically, actually, that's one of the challenges. It's it's also the reason why we're pushing on the Human Immunome Project. It's the sample sizes that we're dealing with in these cases, and not, the larger numbers typically come from these bulk studies, like they just take all the cells, extract the RNA. Those numbers are quite large. But when you look at some of these more cell type specific signals, some may or may not be translatable to the bulk signals, or yet at least. So therefore, we are a little bit limited in evaluating some of these questions in terms of like what are the overlap, what's the context specific correlations that you may see, and based on this on the preliminary work we have done so far, I suspect that these things are not independent at all. I think there's like multiple underlying common mechanisms that are driving these things, but yet we see them as variable and sometimes strong, sometimes weakly correlated. It's because we're looking at biased subsets of people in a way, right? So therefore, under certain contexts, when the variation across people are high, for example, when you look at a huge age range, if those things are linked to exposure amount and history, then you're going to see a strong correlation because of that variation in the age group, for example. So I think we need much larger cohorts and more contextual physiologic variables for us to understand better on the condition, or in technical terms, the conditional distributions, basically, of these signals. Yeah, but I think my feeling based on the preliminary data we have looked at over the years so far, it's that these things are not independent at all. There's multiple underlying, but the root of these signals, I think it's something that we need to work on, basically. Yeah.
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 One last question. I know you're, and we're sort of way over time, but this is so fascinating. A lot of groups are looking at whether or not an immunological signature could be driven by the microbiome. So there's a lot of data on it, a lot of papers. And so how might your tools track that, pick up on that? Is that an easy answer? Probably not.
Not an easy answer. Yeah. So we are starting to do some studies where we collected at least like stool and samples like that. So with trying to answer that question of like, can we find connections? But I may be a little, I think at least on stool, it's a bit tricky because the signals can be it's a function of things like diet, for example. That's a major shaper, right, of what's
Mhm.
going on. And while they're temporally stable signals, they tend to be much less so or stable in a way. If you look at the quantitative dissections, it could be because of technical noise also. Then
But
some
if you're down in the infancy timeline, that you at least you're a little more controlled in some
true,
ways.
true. Yeah, those I think I agree. So we have collected some diapers, but haven't opened them up.
All
In
right,
those,
to be continued.
yeah, to be continued.
To be
Exactly.
continued.
Yeah.
All right, John, I'll try to connect you with some people that might be interesting to you and you to them. Thank you
Cool.
again for
If you don't
fitting
mind
us in.
sharing the chat thing with me so I can review and see if I can respond to some things I have.
Absolutely.
Yeah, or the
Yes.
paper you share. Yeah.
Yes, I'll absolutely share all that. So thank you
Okay.
again. But
Okay. Bye.
I
Take care,
really
Monica.
appreciate it and have a great rest of your day. Enjoy your time in Beinecke. Bye.
Bye bye.